Hello, this is Dr. Smita Narayan Karthik, and today we are going to talk about the very important aspects of the questions to be covered in the plant chapter, that is plant related chapter, that is sexual reproduction in the flowering plants. Yes students, I hope you remember one video nalli naan heliddu, this is one of the very important chapter. ಯಾಕೆಂದರೆ many questions ಇಲ್ಲಿಂದ actually ಬರುತ್ತೆ. And the weightage of the chapter is around 4 to 5 marks. Okay, so now let us start with some important aspects or questions of this chapter, right? Okay, and here we go. The first question is a diagrammatic question, and nimge idu diagram gottirbodu, correct? Second PUC, you have studied this diagram, right? Okay, let us now go to the options. So identify A to E in the following diagram. A, option A, tapetum, microspore, mother cell, middle layer, endothecium, epidermis. Option B is A, epidermis, middle layer, microscopic mother cell, tapetum, endothecium. Then option C is middle layer, epidermis, tapetum, microspore, mother cell, endothecium. Then last option D, that is A, epidermis, B, endothecium, C, middle layer, B, microscope mother cell, and E, tapetum. I hope you little bit remember the diagram of this, that is the stamen. Yeah. Let us now come to this concept. What is this diagram basically? Yes, it is a TS of the mature anther. And let us now what it actually presents. Okay, now imagine now this is one stamen, right? Stamen represents dash of the flower. What is it? Male or female reproductive part? Yes, it is very right. Stamen is actually the male reproductive part of the flower. And yes, each stamen is comprising of two major parts. First is the anther, second is the filament. Now, let us talk about this anther. Now, imagine now this is one anther and we represent it like this. So, here we go with the anther representation. So, imagine now this is one anther and this is the filament, right? Now, we say there's a two anther lobes. So, that is what? Bilobe structure. So, the anther shown here is a bilobe structure having two lobes, right? And if you see, each lobe has got two thecas. Each lobe has got how many thecas? Two thecas. So that means these are the four thecas in the two lobes. That means in each lobe you have two thecas. So it's called as what? Dithecus. What is it called as? Dithecus. So now this is a representation as if you know the anther is the stamen is standing erect. And now we are going to cut the anther in such a way that is going to be called as a transverse section. Transverse section in the yenu students, imagine the plant is standing erect and you take the TS. Now I make it horizontal. So it project nimatra agtade, right? So that is going to be the TS in a horizontal manner, and this is what the diagram shown here, right? So these are the four thecas which was represented here like this, correct? Now let us try to focus on the diagram, that is TS of the mature anther, and here it goes. So first of all, if you make out this thing clear, these are the four thecas, right? Correct? And now we will take one theca and we are presenting it here, correct? Let us now try to understand the layers of that theca. Okay, so if you see, there is the outermost layer called as the epidermis. Called as what? The epidermis. Epi andre yenu? Grammatically, epi andre outer, like epidermis of the skin. Epi andre outer. Dermis andre layer, correct? So epidermis stands the outermost layer, right? ಆಮೇಲೆ ಏನು ಬರುತ್ತೆ ನೋಡಿ, just beneath the epidermis you find something called as what? The endothecium. Endo present inside. So it will be present here, right? Just still go ahead, ಏನು ಸಿಕ್ಕುತ್ತೆ ನಿಮಗೆ? It is the middle layer. ಇಲ್ಲಿ ನೋಡಿ the middle layers which is represented here in this format. So here again you find the middle layers. And next you go further, you get the very, very important nourishing tissue or the cells that is called as a tapetum. So here is a tapetum, right? So this tapetum stands the very important part because it provides nourishment to the cells which are present inside. And what is present inside? and that is the microspore mother cells, MMC. These microspore mother cells are actually requiring the nourishment which is provided by the tapetum. So tapetum can be another MCQ being asked for you. So what is it going to provide? Yes, students, tapetum, the main function of the tapetum is going to provide nourishment to the microspore mother cell. Microspore mother cell ಆಮೇಲೆ ಏನು ಫಾರ್ಮ್ ಮಾಡ್ತದೆ? Yes, it will lead to the formation of the pollen grains. Very important, student. So this is what, as we discussed, so 3 layers, which is actually prime main function is to protect the tapetum, and inside the tapetum you get what? The microspore mother cells. And now ನಿಮಗೆ ಆನ್ಸರ್ ಗೊತ್ತಿರಬಹುದು ಅಲ್ಲ? What is the answer now? Can you tell me? Think of the question and now tell me the answer. What is it going to be? Option A, B, C or D? Here, look at the diagram. A, B, C, D, E, right? And the answer here is A stands for the epidermis, B, endothecium, C andre enu, the middle layers. So can you tell me what was the function of these 3 layers? That was mainly the protection to the innermost tissues, that is the tapetum, and of course the microspore mother cell. And here were the various parts of the anther, right? So this was the first MCQ, and as I said, related to this, definitely we can ask you 4-5 time MCQ connected with this, like tapetum, nourishment tissue, and etc. We'll throw some light on that too later. So this was the first question, and the answer here is option D. Now let us see the next important question, and here it goes. What is the function of the germ pore? Germ pore andre ಏನು? ಎಲ್ಲಿರುತ್ತೆ ಇದು? Okay, let us now try to understand the options here. Function of the germ pore. The first option is emergence of the radicle. Next is emergence of the pollen tube. Next is release of the male gametes. And finally, absorption of water for seed germination. So first thing what comes in your mind is strike out the one which is not required. Before that, I give you one hint, that is a diagram. Okay, again this diagram is known to you. You have studied this in the theory. ನಿಮಗೆ ಇದು diagrammatic presentation draw ಮಾಡಲಿಕ್ಕೆ ಇತ್ತು, ಕರೆಕ್ಟ್ ಅಲ್ಲ ಸ್ಟೂಡೆಂಟ್ಸ್? ನನಗೆ ಸ್ವಲ್ಪ ಮಾರ್ಕಿಂಗ್ಸ್ ಹೇಳ್ತೀರಾ? Okay, what is this outermost covering which is called as? Now look at this, students. Pollen grains are meant to be the very important part of the flower, because pollen grains are the one which will be transferred from the anther to the stigma of the flower, and that will be called as pollination, which will result into the development of the end product, that is the seed, and later that is how it goes further, that is the formation of zygote, and later. So don't you think so? Pollen grain should be designed in such a way that it should be well established and well sufficient, and cannot be damaged so easily when it is been exposed to the environment. And God is really great. He has designed the entire pollen grain in such a way that it cannot be destroyed so easily. Okay, let us come back to that now. So I gave you one hint. Illi ಒಂದು answer ನಾನು ಹೇಳಿದೆ indirectly, and you should be in the position what is that word called as. Okay, come to this point now. So this is what the structure of a typical pollen grain, wherein you get the outer layer which is actually found to be tough layer, right? And that is called as the exine. What's it called as? Exine. ಇದು exine ಮೇಲೆ ಸ್ವಲ್ಪ ಡೆಪೋಸಿಷನ್ ಇರ್ತದೆ, ಒಂದು ಸ್ಪೆಷಲ್ ಸಬ್ಸ್ಟೆನ್ಸ್ ಡೆಪೋಸಿಷನ್. ಅದು ಸಬ್ಸ್ಟೆನ್ಸ್ ಹೇಳ್ಬೋದಾ ನೀವು? ವಾಟ್ ಇಸ್ ದ ಸಬ್ಸ್ಟೆನ್ಸ್ ಕಾಲ್ಡ್ as? Yes, that is sporopollenin. ಇದು ಸಬ್ಸ್ಟೆನ್ಸ್ ಏನಂದ್ರೆ ಸ್ವಲ್ಪ ಸ್ಪೆಷಲ್ ಇದೆ. ಇಟ್ ಇಸ್ ಕಂಪ್ಲೀಟ್ಲಿ ಬಯಾಲಜಿಕಲ್ ಪಾಲಿಮರ್. It is inert chemical, meaning chemically inert, meaning what? Even if you treat this pollen grain with any acids or bases or any enzyme, it will not affect the pollen grain. I mean, the pollen grain will remain intact. It cannot be so easily destroyable also. That's the very strongest part, and we should thank God for gifting the pollen grain with this sporopollenin, which is actually the very strong aspect or asset of this pollen grain. Now let me tell you what is there apart from the exine. So as I said, exine ಮೇಲೆ ಸ್ವಲ್ಪ ಸ್ಪೆಷಲ್ ಡೆಪೋಸಿಷನ್ ಇರ್ತದೆ. ಅದು ಸಬ್ಸ್ಟೆನ್ಸ್ಗೆ ಏನು ಹೇಳ್ತೀವಿ ನಾವು? ಸ್ಪೋರೋಪೋಲೆನ್. ಆಮೇಲೆ ಸ್ವಲ್ಪ ಗ್ಯಾಪ್ ಇರುತ್ತೆ. ಎಲ್ಲ ಕಡೆಗೆ ಸ್ಪೋರೋಪೋಲಿನ್ ಇರೋದಿಲ್ಲ, ಸ್ವಲ್ಪ ಗ್ಯಾಪ್ ಇರುತ್ತೆ. ಅದು ಗ್ಯಾಪ್ಗೆ ಆಕ್ಚುಲಿ ನಾವು ಏನು ಹೇಳ್ತೀವಿ, ಜರ್ಮ್ ಪೋರ್. Correct? Germ pore andre sporopollen absent, right? Now, just go inside, you get the intine and then you get two type of cells here. One will be the vegetative cell, another will be the generative cell. Vegetative cell will try to capture the major portion having more of the cytoplasm, and in that is housed the generative cell. So generative cell ಆಮೇಲೆ ಏನು ಮಾಡುತ್ತೆ ಅಂದರೆ slowly as per the development of, okay, it slowly helps for the formation of the two male gametes which will be later ready to fuse with the egg cell of the embryo sac, that is the embryo sac, the female gametophyte. Okay, so now I gave you some hint. ನಿಮ್ಗೆ ಗೊತ್ತಾಯ್ತು ಅಲ್ಲ ಏನು ಸ್ಪೋರೋಪೋಲಿನ್, ಏನು ಜರ್ಮ್ ಪೋರ್, ಎಲ್ಲ ಪಾರ್ಟ್ಸ್, ಎಲ್ಲ ಫಂಕ್ಷನ್ ಏನಿದೆ. ಓಕೆ, now can you tell me the answer for this question? Emergence of the radicle, emergence of pollen tube, release of male gametes and absorption of water for seed germination. Students, ನಿಮಗೆ 4 option ನಲ್ಲಿ ಸ್ವಲ್ಪ ಐಡಿಯಾ ಬರುತ್ತೆ which are the options which you can reject, okay, or you can strike out. So option A, radical ಅಂದ್ರೆ connected ಇಲ್ಲ, right? So it's a wrong answer. Again go to the next one. Option D, absorption of water for seed germination, again illi connected illa ಏನು, right? So the tie-up is between these two options, B and C, and the correct answer here is going to be the main role of this germ pore is going to be for the formation of the pollen tube, and the answer here is going to be answer B, emergence of the pollen tube, right? So these are the two questions we discussed here. Let us try to relate some of the important aspects of the pollen grain, and here we go. Now you can answer this. Now naan ಏನು ಹೇಳಿದಿಲ್ಲ, it is your time, you are going to tell me now. An organic substance, okay, that can withstand the environmental extremes and cannot be degraded by any enzyme. Yes. Before I tell the option, the answer should be at the fingertip. Yes, students, what is it? I can hear you telling. What is that? Very right, student. Gottaita ala? Okay, what is it? Right now we had studied. Very beautiful, students. The answer here is again the sporopollen, right? As I said, it is resistant to the action of enzymes, the strong acids and high temperatures and alkali also. Thus, a very important point to be noted. It is found to be extremely stable and chemically intact, okay? Extremely stable and intact in sedimentary rocks which date back to 500 million years ago. That means as for evolutionary point of view is concerned, it plays a very important role in the phylogeny too, right? So this is a very important concept about the sporopollen. Rest all, if you see, students, the cuticle, the lignin and the cellulose are actually getting degraded when we treat them with these chemicals, but then except the sporopollen. Before that, I give you one small, okay, a question here. Think of the function of cuticle and tell me where is the cuticle present, where is the lignin present, where is the cellulose present. Salpa alochane ಮಾಡಿ tell me. You have studied in first PUC, ನಿಮಗೆ ಗೊತ್ತು ಇದು lignin ಏನು. ಸ್ವಲ್ಪ think ಮಾಡಿ, and next session I will be back for the discussion, right? Okay, so this was the next question regarding the sporopollen, and here we go to the next question, right? What is it? In fossil deposits, okay, fossil andre gottagbodu answer, correct alla? Pollens are seen well preserved due to the presence of, what is that? Just we have discussed, and right, the answer here, okay, this is again which is not connected at all, again not so with respect to the fossil formation. The tie-up is between these two, right? Sporopollen ಎಲ್ಲಿತ್ತು, exine or intine? Intine alla, right? No, students, it was present in the exine region. So the answer here is sporopollen in the exine region, and the answer here is option B, sporopollen in the exine. So yeah, so these were the very important questions about the pollen grain. Okay, now let us go to the next question, and here you go with the next question. And the next question is, pollen tablets are available in the market. Pollen tablets sikuta? Okay, let us discuss why and how. Okay, pollen tablets are actually available in the market, and yes, they can be used for in vitro fertilization, breeding programs, supplementing food, that means as food supplements, and then ex situ conservation. Acha, that means pollen tablets, how are they made? Yes, students, it is found that the pollen grains which we discussed right now are rich in nutrients, like they are found to have proteins and vitamins also, which can be a great source of supplementing, that is a food supplement. In abroad, many places athletes consume this, as well as even animals are being fed with this pollen tablets, because it has got high source of energy too. So that's the reason in many modern countries pollen tablets are being formed, which is actually made and for consumption. So this is the answer here. The answer here would be supplementing the food. So as we discussed, the pollen contains the important element for weight enhancement, and yes, very important aspects. It helps to enhance the immunity and some inhibition of the respiratory infections also. Product obtained such as are available in many forms, so like you can get syrups or tablet form also. So as I said, this is a very important, nowadays growing as a very good supplement in food, right? Okay, so now we go to the next one, and here the next question. Pollen tubes show chemotactic movement, thermotactic movement, chemotrophism, chemonasty. Now this is something very important, okay? There are many terms in the options, okay? So students, in the previous standards or in first PUC, you must have heard about trophism, right? So what is this trophism? There is something called trophism, tactic and nastic movements, right? I give you a single description in between and difference between all the three, okay? Trophic movements andre yenu. Trophic movements andre, suppose now this is the seed, okay, and this is the radicle, this is a plumule. We say the radicle goes towards the soil, plumule goes towards the sunlight. So when we say the part going or part growing towards external stimuli, okay, growing towards external stimuli, and the direction will be specific. It says directional specific. So that movements will be called as trophic movements. So whenever we say trophic movements, it is going to be directional. It's going to be directional. Like roots are positively geotrophic and the shoot is positively phototrophic, like that. What is this tactic movement then? Yeah, very important. Tactic also is movement only to external stimuli, but there's a difference. Tactic directly translocates the organism from one place to another. That means there is change in the place. For example, algae. So algae is the best example which we can give here, moves towards the source of light. In water body, algae tries to capture sunlight for photosynthesis. So movement of the single algal cells, literally movement, will be changing of the place, will be called as the tactic movements, right? And next is nastic movements. Like, you must have heard about the Mimosa pudica plant touching the leaf and it droops down. So these are called nastic movements. Also, a climber, if the climber wants to take help of a wooden plank or something, it will just tap or hit randomly to the surface or the support, and it will try to surround. So that movements all, which is not again directional specific, it is called as what? The nastic movements. So here I can say there is completely a translocation or transportation of the organism from one place to another. That will be called as tactic movements, like algal movements is a best example. Then comes the trophism. Trophism meaning the movement is going to be directional, like the roots are going towards the soil, positively geotrophic and negatively phototrophic, etc. And nastic movements are again the movements, yes, which will not be directional. It can be in any direction. So these are the three important aspects about non-directional. As I said, I just write here non-directional. So these are the three important aspects about what the movements, right? And yes, they can be with respect to the external stimuli like heat, light, etc. Okay. So now can you tell me what is this pollen tube going to show? So what can we connect with this? Nan just helide nimge pollen tube actually elli form agutte? Yeah, from the germ pore. Germ pore inda adu generative cell actually two male gamete form ಮಾಡುತ್ತೆ, correct ಅಲ್ಲ? So ಅದೇ ಇದು concept. So come back to the diagram here. This is the entire stigma, style and ovary, and this is what the ovule, right? And inside the ovule you find the presence of the female gamete, that is what the egg cell. So egg cell would be present here, and now it is a time for the pollen grain. Now look at this. Already the pollination has taken place, and now the pollen grain has to travel from the stigma and reach to the egg cell. So slowly the pollen grain will form the pollen tube. And yes, the pollen grain now is actually just shed, and slowly this pollen tube starts forming now. So pollen tube will be start forming, and slowly it will help for the two male gametes to be formed, and slowly the male gametes will be transferred here. So that movement, you know, where there is a sugary substance present to be found at the stigma. So it is found that stigma nalli actually sugar substance irutte. So once the pollen grain sits on the stigma, the pollen grain gets activated by the sugary substance, and then it starts slowly moving towards the respective destination. So the answer here, pollen tube show what movement? That is going to be called as what? The answer here would be chemotrophism. Yeah. Okay. Now the next question, the very very important question. Again, it's a diagrammatic presentation, and new namge helbeku illi yen option barutte illi. Okay. Have you heard about antipodal cell, egg cell, synergid, zygote? Which part of the plant is it? Yes, think about it. That is the embryo sac. Okay, let us now come to that. Out of the 7 names, find out the haploid. Haploid andre yenu? Haploid andre N. Correct? Haploid andre N. Right? Diploid means 2N. Triploid means 3N. So now you are supposed to identify out of these cells which are present in the embryo sac, which are haploid cells, right? That is N, single set of chromosome. N stands for. Okay, and here are the options. Before I give the options and the answer, you will describe me this diagram. It's a diagrammatic presentation of an embryo sac, and I hope you know how is the embryo sac formed. Yes, it is formed from the megaspore cells. Now, how it is formed from the megaspore? Yes, students, megaspore cells actually are formed 4 in number. Out of that, 3 cells degenerate, and 1 megaspore left out cell will lead to the formation of the embryo sac. And yes, the one single megaspore which is going to lead to the formation of the entire embryo sac is actually haploid. So that means the megaspore mother cell which will be leading to the embryo sac will be actually haploid, and it is going to show mitosis. Mitosis is an equational division. Mitosis is a equational division. So there will be series of mitosis happening in such a manner that all these cells are slowly being formed. Initially there will be free nuclear division, wherein the cell won't be formed. Only the nuclei will be present. Later you will find that the cells are formed intact in such a way now it is eventually leading into a 7-celled 8 nucleated structure. 7-celled 8 nucleated structure. So can you count? It is 7, 2, 3, 4, 5, 6, 7 cell, and there are 8 nuclei. 1, 2, 3, 4, 5, 6, 7, 8, right? So that is a 7-cell 8 nucleated structure. So this is briefly the outline of embryo sac, wherein you will get 2 ends. One is the chalazal end, one is the micropylar end. So chalazal end will have 3 cells, okay, nucleated 3 cells called as antipodals. Called as what? Antipodal, and they are going to be haploid. Correct? Next comes the cells towards the micropylar region. So towards the micropylar you find the egg cell and the synergids, correct? Again the synergids are haploid and the egg is also haploid. Egg is nothing but a female gamete, right? So it will be definitely haploid in nature. Understood now? So idu haploid irutte mattu idu synergids also haploid, antipodals haploid irutte. What is present now here is the primary endospermic nucleus which will form after the double fertilization. So PEN is going to be triploid. Nucellar tissue, that is a nucleus, is nucellus is going to be diploid. So overall if you see which one of them will be the haploid cells, yes, from this you can make out this is going to be haploid, that is the antipodal cells, then the egg cell, then the synergids, right, and then left out is the male gamete. Okay, so now tell me the option here, which one is the right option here, and your answer is correct. That is answer C, that is 1, 2, 3 and 5. 1, 2, 3 and 5. Things clear now? So what was this very important aspect? This was the structure of the embryo sac which had various 7-celled, that is 7-celled 8 nucleated structure. And now we move on to the very important question, and you are there to answer for this. What is this nutritive function best seen in? Options are egg cell and synergids, which we studied right now, endosperms and the tapetum, which we studied. Tapetum, where did you study tapetum? In the male, that is what the pollen grain structure, right? Not even the pollen grain, that was the stamen structure. You studied the tapetum, and endosperm you studied right now. Male or female gametes, endosperm and the mitospores. Now from this you can make out which one of them is going to be providing nourishment, and you're very right, that is endosperm, and tapetum is going to serve the purpose of providing nourishment, right? Okay, let us move on to the next concept, a concept based on pollination. Yes students, can you tell me what is pollination? Pollination is a very known concept to you. Transfer of pollen grain from anther to stigma. Transfer of pollen grain from anther to stigma. So there are various agents involved, etc., etc., that we'll be studying in the regular sessions or the crash courses. But let us now try to understand what is this dioecious flower. Dioecious meaning sexes are separate. Sexes are separate. Now you will tell me what will be found in the case of dioecious plant. So I already have given you a hint. Sexes separate irutte dioecious plant nalli. So let us now see the options. Autogamy and xenogamy, autogamy and geitonogamy, geitonogamy and xenogamy, clistogamy and xenogamy. Now, before we try to solve this question, let us throw some light on the different types of the fusion of the gametes, right? And here we go. So look at this now, the types of the pollination. So you have studied there are two types of pollination. One is the self-pollination, one is the cross-pollination. Now what is self-pollination? Can you make out from the diagram here self-pollination? Self-pollination andre one plant nalli one flower irutte, correct alla? And you know very well, flower nalli male reproductive part and female reproductive part, that is the stigma style ovary irutte and the anthers also irutte. If it's a bisexual flower, definitely the same pollen grains will be shedded onto the same stigma, right? So definitely that will be called as self-pollination. Within the same flower, the shedding of the pollen grains is taking place, indicating what? Self-pollination. Mattondu type self-pollination also irutte. Andre, let us now talk about that. Same plant nalli 7 flower irabaudu. Correct? But the position would be different. So even if same plant nalli 7 flower between pollination agadre, even that will be called as self-pollination. Got this point clear? So what is cross-pollination then? If the pollination is taking place between two different flowers of two different plants. Okay? Two different flowers of two different plants, then only it will be called as what? The cross-pollination. So there are different terminologies given for that, and here we go. Self-pollination, we basically one type called as autogamy. Now look at the word. Gamy means marriage. Gamy means what? Marriage. And auto means self-marriage. Meaning auto means self. So here you can tell autogamy means pollination taking place within the same flower. Same pollen grains will shed onto the same stigma, right? So here it has been indicated. Correct? Autogamy. Next comes the geitonogamy. Geitono means neighbor. Neighbor. Okay? So pollination taking place with the neighboring flower of the same plant. So you can make out the plant is same but two different flowers involving in the pollination, that is transfer of pollen grains or stigma to the anther to stigma, and that will be called as what? Geitonogamy. Pollen transfer between flowers of the one individual. Correct? So these will be types of the self-pollination. Now go to the cross-pollination. Xenogamy. Xeno matlab stranger. Xeno matlab stranger. So marrying with the stranger. Marrying with a stranger, right? Yes, that means there is another plant involved here. So there is two different flowers from two different plants, and a pollen transfer between flowers of different plant individual or varieties will be called as what? The, yes, xenogamy. Now here is a question again. Can you think of the answer here? I am telling what would be happening in the case of dioecious plant. I mean here they are telling prevent both, which will not happen. Yes, that is what? Autogamy and geitonogamy, because these are the types of the self-pollination, and this is the one where the sexes are separate. That definitely they will be showing the cross-pollination only. Okay, so here the answer here would be answer option B, autogamy and geitonogamy. Next question, emasculation is achieved by. Now what is emasculation? Look at that, and here are the options. Now imagine an example wherein we want to create a plant of our own interest, or you can say there is one plant A, okay, and there is one plant B, okay, and you know there is one plant C, correct? This will have its own flowers, this will have its own flowers, this will have its own flowers, okay. Now see such a plant, the flowers are having such characters which we don't want actually. So we want to neglect this plant or the flowers. We don't want the pollen grains of this flower to get spreaded onto the flower which we are desiring for a new variety to develop, right? So we will try not to allow the pollen grains from this plant to go to other plant. So forget about this plant now. So now we don't want this plant, that is plant C. Okay, so we have a very beautiful flowering plant, that is plant B, having the flowers with it, and yes, we found that there is something, a new variety or a desirable character. So this plant has got some desirable character which we want it to be incorporated into this plant. Correct? Imagine this is an inferior type of plant and we want the characters to be coming from the plant B to plant A. So yes, what are we going to do? The problem can be plant A definitely will have its own stamens and have its own stigma, correct? Imagine now this is the stigma, style ovary and the stamens, and the plant B will definitely be a bisexual plant. Even this is a bisexual plant. The problem with plant A would be, since the plant is bisexual, there are chances that its own pollen grains will be shedded onto the stigma, and we don't want the product of this flower again because it is going to produce an inferior quality of progeny. We want a better quality of the progeny, right? So what are we going to do here is, we will try to cut this anther with a forceps. Correct? The process of cutting or removal of this anther will be called as emasculation. Correct? So once we cut the anther of the desired flower, okay, definitely the point of contamination of the stigma with its own pollen grain is been inhibited now, right? It will not provoke because we have removed the anther. So before this development only, we are going to remove this anther, and definitely now this is going to be made into a unisexual flower, and now we want the desired character to be incorporated from B. So we'll try to share the pollen grains from plant B to plant A, and then this plant A will try to conceive the characters, that is the desirable characters of plant B. So that is a type of artificial hybridization. So this is a very important concept of artificial hybridization, right, wherein we try to create our desired characters into the plant which is having inferior quality. Okay, so this concept is very much involving the technique called as what? Emasculation. So can you tell me the answer here? What is emasculation? Read the options. Removal of stigma, removal of sepals and petals, removal of anthers, removal of gynoecium. And the answer here is going to be, think of the options, and the answer here would going to be what? Tell me, what's the answer here? Yes, the right answer here is going to be removal of the anthers. Okay, so this was what the very important question again. And now this is something which I leave you for homework. Salpa new homework ke liye try maadi. Very simple question. The question here is 60% of the angiosperms shed their pollens at a 2-cell stage, at a 3-cell stage, at the 4-cell stage, and 1-cell stage, and you are going to think about the answer. Okay, so here we try to complete with all the important MCQs, and we'll be there with many more such questions, students. So here, students, we have done with some of the very important questions of the sexual reproduction in the flowering plants, and here is some interesting news for you. You know that what it is? Yes, here at Diksha we are going to have a crash course for you all. Isn't it a very beautiful news for you? Yes, students, this crash course will be held for a duration of 45 days, and yes, it will commence after your board exams, and it will be for residential as well as non-residential students also. And now look at some of the toppers, achievers of Diksha, and here we go. Ronak with 2nd rank, Pallavi with 3rd rank, 5th rank Siddhartha, and Vaishnav with 6th rank. If you want to be amongst these students, the best students, or if you want to get into the best premium colleges, you have to join the crash course. Yes, student, at the crash course we'll be giving you a good amount of the practice which will help you to score well in your exams. And here is a registration number. Please call out on this number, that is 1800-102-4109. Dial this number and register today itself. And yes, students, very important thing to be noted here is, please click the link given below. It will give you with all the information. Students, please do not forget to like, share and subscribe to our channel, and please press the bell icon so that you get further notified also. And please share this session to your other friends, students also, who would definitely be getting some help out of it, right? So here Dr. Smita Narayan Karthik signing off for today. We'll be right back with our next session. Bye then. Thank you, students.